here coming back home to Michigan now, and Tim Cernak, who is a professor in the chemistry department. I mean, we just had a major component of enabling and is the data arena and the data developments. Another is the whole automation of laboratories, which is an important component to the ARW vision. Go right ahead.
Thank you so much. Thank you so much, Dan, for that kind introduction and for the opportunity to speak here today.
Are you on?
In shuffling around. Okay. Thank you all so much for this opportunity to share our research. And wow, these are big shoes to fill following Alex. That was a pretty incredible talk. And my talk will have a lot more of the feel of an assistant professor trying to get tenure and be a little bit more focused on organic chemistry. But we are deeply interested in everything data and chemical synthesis at the interface of data science. And right, so I run an experimental lab in the next building over, and our focus is chemical synthesis. When we started at the lab 5 years ago, we had a component of data science that was we were unique at the time. I think in the NASEM report that's come out, one of the things that I was griping about at that time was that organic chemists weren't learning about data science. And I think that in some ways the pandemic helped us out just a little bit, in that most organic chemists were stuck on their couch for 3 to 6 months or a year, and many took up Python in that period. So that was an exciting time. I should also say it's been, the NASEM report was my very first Zoom meeting, and so Ilke and Tapio, who had been organizers, and Alex and Dan as well, it was, I was so sad because I was really excited to go to Washington, D.C., and I had to download Zoom for that meeting. I was like a WebEx and Skype and Blue Jeans guy in those days. But so it's really exciting to meet you in person and to have you here in Ann Arbor. Thank you for coming. So I actually pulled up the slides that I presented in that NASEM meeting, and the focus was on what are molecules and what is chemical space. I'm going to get pretty deep into molecules, and so I'm hoping that most people in the audience can appreciate that these are molecules. These are antiviral molecules. It was timely then, and I guess it's still timely now, to talk about antivirals. And one of the important things to recognize is that these are, we learn how to draw this at the chalkboard when we're taught organic chemistry early on. You recognize that there's carbon bonds wherever there's a line, and then if there's a different atom, we would actually draw it out like an OH here, and then it's assumed or it's known that there's a carbon-hydrogen bond at all these other spots where I don't draw it in. But so, most chemists would see this and you recognize what it is. A computer sees this and it doesn't really know what it is. Or this is kind of like an artisanal drawing, right? So computers like to see things in this format. This smile string down below is one way that we can communicate this structure into the computer. There's many ways to do this. Or you could just call it its name, right? Remdesivir. So, maybe there's a relational database that says this is remdesivir, and it looks like this if I draw it on the chalkboard, and it looks like this if I want to put it into a database. The important thing that we'll get into is that molecules are graphs. Perhaps a lot of people in the room are well aware of this. I was unaware of this, having studied organic chemistry for 20 years, and then kind of became aware of it through an interest in data science. And it's a very old field of research that molecules as graphs goes back to. We've been studying papers from the 1870s where I think that graph theory was important to the understanding of molecules and how they could be generated. And so here's oseltamivir. This is a flu specific molecule, and I show here a synthesis of oseltamivir along the bottom. Hopefully this looks a little bit more graphy to you than this chemical structure, but they're exactly the same, right? So I got black dots and red dots and blue dots to show carbon, oxygen, and nitrogen. An important thing to recognize is that there's chemical reactions that we would use to stitch together this molecule. So oseltamivir has saved many lives, perhaps millions, I actually don't know. But at one point, a synthetic chemist was involved in making this molecule. And as I'll probably come back to a couple times throughout this talk, there's perhaps an infinite number of molecules that could exist. And so, one chemist needed to say, I think that this molecule, this product, might have value to the world, and we'll try to make it. And then they embarked on a synthetic quest, which here took 5 synthetic steps, 5 different reactions shown as these little orange circles to stitch this molecule together. But they really could, given an infinite number of molecules, they could have picked any other one. Each one of these chemical reactions is likely to fail. I don't see any of my students here, but we fail a lot. Like when we run experiments, probably 99% of what we do, 99.9% of what we do, well, 98% of what we do fails. And you just get the data, it didn't work. That took me 48 to 72 hours to get that data, and I just throw it in the trash. So most of what we do fails, and so you need to be very careful about what you decide to make. And so it's interesting that some chemists decided that oseltamivir could be an interesting molecule, and they tried to navigate through all that failure to stitch together these 5 reactions and actually make the molecule, and millions of lives have been saved on the basis of that. But new molecules are needed to do many things, and each molecule requires multi-step total synthesis. So we have to stitch together these sequences of reactions to take the chemicals that we can buy. There's like 67 million molecules that I can buy from vendors right now, and then stitch them together through a sequence of chemical reactions that are known, and make any of this infinity of chemical space. That's going to be our next agrochemical material, perfume, or medicine, is going to be some molecule with a shape. And thinking about these molecular shapes is interesting, but one needs to invest in the risk of actually making the darn thing. And so these principles of I write this as a medicinal chemist, but this applies to really anything. I think have advanced a lot in the past decade, and makes a lot of sense in terms of chemical synthesis. And so we would design a molecule. That's using the graph to stitch together the chemical structure that I think is the right chemical structure. It's a design based on data that exists. Maybe Professor Chalet talked about active learning, and we're definitely excited about the notion that that design would be informed by the making and testing of the molecule before it, and you could imagine some active learning loop that is designing new molecules, making the molecules, and testing them. And so my specialty is making the molecules, and the interesting thing is that what you make, or what you design rather, is informed by what you know you can make, right? We love to propose things that we don't know if we can make it, and then we'll try. And I'm going to end this talk, if I get there, telling you about a molecule that we spent 4 years trying to make. But we wanted to really try to make that molecule in an industrial context. That's not going to work, right? I mean, typically in an industrial setting, you've got 1 week to get your molecule in hand and learn whether it's good or not. So you need to really de-risk this making step. And then for a medicine, we're steering towards something like this, an objective shown here as a Venn diagram, where your molecule is the black dot in the center. And this has the balance of solubility so that you can swallow the pill and it dissolves in your stomach, and permeability so it gets across your intestinal barrier into the blood, selectivity so that it doesn't have toxic side effects, and metabolic stability so I can take 1 pill a day, and potency so it does what it's supposed to do. It treats cancer or Alzheimer's or COVID. And then as a synthetic chemist, I kind of greedily say that it needs to be synthetically accessible. We need to be able to make it, or it just won't come to existence. And hopefully that's straightforward. Okay, so this is my one background slide on technology, and so this is pretty deep for organic chemists, but here we are. There is a large field of organic chemistry called total synthesis, where we try to make new molecules. We try to stitch them together. And over the course of about 15 years, a professor named Bartosz Grabowski had his entire team encoding the literature manually. This is now done with machines, but there's a lot of expert encoding of rules of chemical reactions, and just one of these rule sets is shown here. So here is our chemical reaction that takes this graph and adds an oxygen, and then the reaction occurs and it makes this set of products. It's called an enantioselective A3 coupling reaction. Here's the mapping of the atoms, this reaction SMARTs. And there's protecting groups that we might apply. We actually don't like to do protecting groups because they're a little bit of a band-aid and they require extra effort that we'd prefer not to. But these are functional groups in this reaction that would need to be protected. And then there's incompatibilities. If somewhere else on the molecule I've got one of these functional groups written here in these SMARTs strings, that reaction is not going to work. It's likely to fail. Okay, you know, a large research group over 15 years in Poland encoded the literature in this way, giving us 100,000 rules. It's become a commercial software by Millipore-Sigma, and full disclosure, I am funded by Millipore-Sigma. I'm going to talk to you about their software. But this is some background in this space that there's a encoding of chemical synthesis so that we can stitch together these multi-step pathways in this way. And what we get is these sequences. So here is a computational retrosynthesis of a molecule, which will be this yellow dot in the center. And then this goes out into these intermediates, which are these different colored dots terminating in commercial starting materials. And so SYNTHIA is the name of this software. And a bit of my story today is about how this is a starting point for retrosynthetic analysis. We've been using this in a slightly different way. And so when COVID hit, Sigma Aldrich, or Millipore-Sigma, was they're the leading supplier of chemicals to the world. And they were very nervous that they were going to get a phone call soon for 1 billion doses of some drug. This is in March of 2020. They didn't know what chemical they were going to be asked for, but they figured, like, we're going to have to supply a lot of something, and we don't know what. And so they asked us to use their software to look at 20 drugs that were in clinical trials in March of 2020 for COVID. They were very concerned that not only would the drugs run out, but the starting materials to make those drugs would also run out. And so we had one heck of a time in March and April of 2020, running this experiment to find alternate supply chains to 12 different COVID drugs that were under evaluation for in the clinic at that time. We were able to open our labs. The University of Michigan would not allow one of my postdocs to drive from Detroit to here, because if you recall, Detroit was an epicenter of COVID. So they said he has to move to Ann Arbor. So we had to move him to Ann Arbor. And it was just one crazy 8 weeks of my life. We had the rest of our team on Zoom or I think we used Blue Jeans actually in those days, around Ann Arbor and actually around the country, crowdsourcing the literature. And they would scrape out every patent, every publication that was known for one of 12 COVID molecules and go through the route and find the starting materials that were purchased to make those drugs. So it's like pieces of Lego, and you have your Lego starting pieces and you kind of build up, right? And it's like, okay, so let's find all the red pieces of Lego that have been used towards remdesivir. And these can be then encoded as these SMILE strings. It's a string of text. So our team crowdsourced the data, and through this automated software, we were able to say, give me a route to this drug, which is undergoing a clinical trial for COVID, the yellow drug in the center, that terminates in a starting material that I can buy, the pink dot up top, and it's not the orange dot, which is so we just had the orange dots were the commercially viable routes or starting materials. That became our exclusion criteria. And so we found alternate supply chains to 12 potential COVID drugs with this approach. And then we went into the lab and had a team working on this. And so here is galidesivir. This turned out not to be a relevant molecule to COVID, but in April of 2020, that was unknown. And so you can see that there's a pretty dense patent literature around galidesivir from this pretty deep network of routes. Each one of these routes is terminating in a different series of starting materials, different Lego building blocks. And so using the SYNTHIA software, we were able to back calculate to different starting materials, which are shown here, and go through a sequence of reactions, which I won't get too much into the details for this audience, but these are pretty cool reactions that stitch this together. And one thing you can note is that our proposed route is, kind of roughly about the same length as the commercial routes. So luckily, we didn't end up in a situation where we needed to supply galidesivir for the world because supply chains were navigated. But we did this for 12 different compounds. Each of these were drugs that were being tested in the clinic for COVID at that time. And hopefully you can appreciate the data challenge that exists here. So there's this long-standing notion of combinatorial explosion in our field, that we have these graphs and carbon gets 4 bonds, nitrogen gets 3 bonds, oxygen gets 2 bonds. There's a lot of opportunity you've got based on just those rules. And so with the 67 million commercially available building blocks, getting from the target that you want, which is some new complex molecule, back to those simpler commercial materials is a little bit challenging. So this was a fun data challenge science for us, and we actually learned quite a bit. Everything that you see here is computational, and so this was then performed experimentally. The 2 very brave postdocs navigated this. Millipore Sigma supported the work, and part of our contract with them was that they had to provide us N95 masks, which in like the second week of March in 2020 was like a pretty big ask. They scoured the world and they found us a box of N95 masks that were expired in like 2007, but they're like, this is the best that we can do. Anyways, this was just an incredible week. The 8 weeks rather, the 2 postdocs who did this work had to carry letters in their pocket from our dean in case the police should stop them and say, hey, you're not allowed on the street. And they had like, I'm doing like critical research. It was a lot of, it was fun-ish, but I was honored by my team in these days. And so they took this network of possible sequences to UMIFENOVIR, which was a drug that had been trialed for SARS-CoV-1. We thought that this could be particularly important because it was a SARS drug. It was for SARS-CoV-1, and we thought that maybe there'd be some relevance. This was the only coronavirus drug that was known at the time, and so other than remdesivir, rather. So we were very excited that we were able to exclude all of these known commercial routes and come back to very cheap starting materials to get there. So this was one big data challenge that, in chemical synthesis. Now, an important thing to note is I told you that we have these templates, these 100,000 rules that are baked into the software that 15 years of Polish PhDs are based on. And these are based on known reactions. They're de-risked reactions. They're reactions that we know will work, right? I mean, we want the computer to propose something that hopefully will work, and that's why there's incompatibilities baked into the rules. But these are the basis of known reactions. And so I just want to pivot a little bit and tell you a little bit about how we're thinking about reactions. And so, writ large, we focus on chemical synthesis and data science. I show you here a big flask, and what we're actually known for is reaction miniaturization, which I won't get too deep into today. But I want to talk about known reactions. And it turns out that there's some really popular ones. There's some that work really well. And yet, I mean, if I were to scour the literature, probably today, since we started this meeting, there have been 12 new reactions that have been published in the chemical literature. So there's like all these reactions that are out there, and yet we gravitate to just a small handful of them. And so particularly in the drug discovery industry, this is the medicinal chemist's toolbox. Before coming to Michigan, I spent 10 years working in the pharmaceutical industry, and I became very familiar with these particular reactions. And so what you see here is a summary by my friend Jonas Bostrom of patented literature and medicinal chemistry publications, where they have scraped the medicinal chemistry literature and patents and said, what reactions do medicinal chemists run? And the really intriguing thing that you see here is, there's one reaction is the amide coupling, okay? And it makes sense that would be a good reaction because the proteins that we typically interact with when making drugs are amides, so there's maybe a little bit of complementarity. The next one is the Suzuki coupling. That won the Nobel Prize in, I think, 2012 perhaps, or 2011. And then the Buchwald-Hartwig coupling. I'm I really believe this should win the Nobel Prize very soon. It's the only new reaction in my lifetime, or reaction generated in my lifetime, that's made it onto this list. There's reductive amination, or amine electrophile couplings. And then the fifth most popular reaction in a $1 trillion industry is this Bhakti protection, which is not even really a reaction. It's just like a kind of like pulling off like a little bit of a Band-Aid at the end of your sequence. And then we get into a diversity of reactions, right? But in a $1 trillion industry, a single reaction dominates 25% of opportunity here. And so this means that 25% of the time we're making molecules that have a certain flavor to them. And this was the question that we posed when opening our lab, is like, why is this the reaction that people would run 1 out of 4 times in drug discovery? And even beyond that, why is this even the reaction that one would run when presented with an acid and an amine? So we'll look at this a little bit differently. Here's a simple amine. Okay, so amine means that it's got a NH2 and a simple carboxylic acid. So this is ethylamine and this is propanoic acid. And the amide coupling forms a bond from this nitrogen to this carbon, and it kicks out water, and we get this amide product. Now, this is that reaction that's run 25% of the time. And if we look at the properties of these molecules, there's something kind of interesting that becomes apparent. And this was something that hadn't been looked at too much before we asked the questions. So this is a basic molecule, and this is an acidic molecule. And they unite to form a neutral molecule. And this neutral molecule it's a little bit polar. It's got what's called a hydrogen bond donor here. So this can form hydrogen bonds within the protein structure, and a hydrogen bond acceptor here. And so that's quite relevant. When we talk about inventing medicines, we care about how many hydrogen bond donors, how many hydrogen bond acceptors there are, whether things are basic, whether they're acidic. This is going to, these are the properties that companies like Pfizer and GlaxoSmithKline and Merck are going to use to determine whether their next medicine is getting into the brain, going to the liver, going to a tumor cell. You look at these really simple properties as like, is my stuff basic? Does it have lots of hydrogen bond donors? And so we thought, okay, if there's this super popular reaction and it has this property of taking a basic molecule, an acidic molecule, and uniting them to make a neutral molecule, what else could we possibly do? What if instead we tried to do this? So this is a reaction that doesn't exist, but there's nothing physically wrong with it. It's just that we haven't found a recipe or a catalyst that would enable this piece and this piece to unite and make that bond. But the cool thing is that this bond is basic. Okay, so this is a basic amine. So if we could find a catalyst to take this and this and turn it into this, we could have a way to make molecules that would be more able to get to the brain than molecules that look like this. Now, of course, I'm drawing just methyl and methyl here, and there would, in the drug discovery context, there'd be like bigger stuff out there. But this is just the reaction that would unite my 2 pieces of Lego, for instance. And so, maybe there could be a reaction that does this, that breaks a CN bond and makes a CO bond. And so this unit is almost the same as the amide. The only difference is that this NH has been swapped for this oxygen. And so this is relevant. These molecules, and we have shown this, are more permeable. They're more able to get across cell membranes, and particularly the blood-brain barrier. And so, we actually do have a catalyst that will do this reaction. And so, our dream is that we have a catalyst that when performing a reaction, you would make your molecule more readily available to get to the brain, or more easily available to go to the liver, depending on what type of disease you're trying to target. And maybe there's a way that you could deaminate and decarboxylate and make a carbon-carbon bond. And so I'm showing you three reactions. They don't exist, or they, in the past several years we've developed them, but they didn't exist. And recall that this is the reaction one would run when performing an amine-acid coupling to make this amide bond. And so we were intrigued that there's three reactions that don't exist, and it turns out that there's many more. And so in the same way that we would classically go to our new box of Lego and get pieces of Lego and stitch them together and say, okay, I've got a medicine that is that is, not getting into the brain. I want it to get into the brain. So I'd go to my big boxes of Lego and I would say, get me the pieces that have fewer hydrogen bond donors, and I'd unite them in a single way. And that's that's our status quo. But we kind of said, what if you had like only 2 pieces of Lego? What are all the different ways you could bring them together? These would require reactions that don't exist. And that's the kind of secret sauce to what we're doing, is that, that's nonsense. You can't propose something because you'll never be able to make it. But we believe in the not too distant future, you'll be able to predict how to make a new reaction out of thin air, or from data and from a lot of the machine learning algorithms that probably many in this room are developing. And so here is shown some complex molecules where we took a complex acid or a complex amine. These are actual drugs, and then we diversified them using a simple amine or simple acid, and we can make the amide. We can kind of move this piece over to here. We can reduce the products. We can make esters. We can make CN bonds, and so we can change the properties of our molecules. It's going to take us a long time before we'll be able to publish this paper, but we've done this on microscope stains, and so we have a single microscope stain that has a carboxylic acid off of it. And we've made 12 different analogs with different catalysts, and it changes the properties by that kind of like Lego combinatorial stuff. And then that microscope stain, because it glows, I can give it to a cell, and so we have one catalyst that will make it go to the Golgi, one catalyst that will make it go to the lysosome, one that will make it go to the nucleus, one that makes it like not get into the cell at all, and hit these kind of different components of the cell. And so the fun thing is that we're doing that with the catalyst, and that's kind of the new part. That normally you'd go get new pieces of Lego. We're saying, if you got these pieces of Lego, can you find a recipe to generate molecules with different properties? We also find that this reaction agnostic approach is really good for retrosynthesis. So I talked about how we would take a complex molecule, here's sitagliptin, and try to break it down to commercially available starting materials using known chemistries. We would focus on those robust chemistries that we can trust and we know they're going to work really well. And in our lab, we like to say, well, forget trusting it, forget knowing, like it's better if it's unknown, and then we'll figure out a way to invent it. And so here is sitagliptin, is a multi-billion-dollar diabetes drug, and each one of these purple lines represents a new way to disconnect it by amine acid couplings. These are reactions that do not exist. This breaking sitagliptin down into this piece and this piece would invoke a reaction that doesn't exist. But these are quite reasonable ways to break apart this molecule. These would, if we could make that reaction, these would be good ways to make this. And so, all right, so you need to, the experiments are tough, experiments fail, and so one of our specialties is miniaturized experiments that we do in a high throughput format. We use robotics for this. We have mosquito robots and OpenTrons robots are the main ones that we use. And we have a really fun software for driving all of this, and it's totally free for use. So we invite you to check out factor.cernaklab.com and play around with the software. And of course, we do machine learning. Here I see Ambuj Tawari's in the lab in the house, and he's been very instrumental in developing strategies to take the data that we're good at generating and learning what to do next. And so for right now, I don't even have a slide on it, but we're really good at making like 1,000 data points. That's kind of where we are right now. And so we were approached by SCHMIDT Futures and said, can you make this open and fair and freely available data? But we want you to pump it up 2 orders of magnitude. And so we're right now trying to build out the lab so that we can give out hundreds of thousands of reaction data points. I was really kind of floored by the last talk. At the end of our 2-year project, we will maybe breach 1 terabyte of data, which to us is a lot. But it's right now we work in megabytes. And so anyways, it's a very old field. Okay, I showed you our algorithm for taking this amine and this acid and making the amide bond. And incorporated in that simple Lego algorithm was things like this, where we do a coupling. So this is a cool reaction that doesn't exist. We could maybe couple and fragment, and that would be a new cool reaction that doesn't exist, and then we could reduce. And so these were incorporated in our first algorithm, but we intentionally left out cyclization because we immediately hit combinatorial explosion, and it was too hard to think about where to go from here. So we left cyclization off the map. And then as we started poking at this, we realized we actually left a lot off of the map. So these 6 possible transformations, they don't break any laws of physics. It's just that they're kind of challenging to imagine how you'd ever have a catalyst that does them. So we left these off. But we said that there were 320 different amine-acid reactions, and the amide coupling was the only one that was known. So and that's the one that is 25% of a $1 trillion industry. And so what if you could do these other things and make molecules with different properties? But we wanted to figure out how many reactions are there? Was a question that hadn't been asked. And so I show you here ethylamine and propanoic acid drawn as graphs, and this this is, we haven't even published this in a peer review yet, so this is about where my skills top out in terms of graph theory. I think that hopefully for like 10 people in the room, this will not be like child's play. But okay, so here's we have 8 heavy atoms. We have ethylamine and propanoic acid. And so because there's 8 heavy atoms, we make an 8 by 8 adjacency matrix. You only have to look at half of it, of course, but we just draw it symmetrically. Wherever there's a cross peak, that's where there's going to be a bond. So I see a cross peak between 1 and 2, and so there should be a single bond between 1 and 2, and yeah, I see it right there. And then I see a double bond, the orange one, between 5 and 7, and yeah, okay, I see the double bond is there. So that's the starting materials, and then the product looks like this, where we have, it looks like there's a bond broken to 8, and so yeah, so 8 doesn't have anything connected to it, and here I can see that there was a bond from 5 to 8, and that's the water getting kicked out. So then what I can do is I can take my product graph, and I can take my starting material graph, and I can subtract them, and I get a transformation graph, and that tells me what the reaction could look like between an amine and an acid. And so the fun thing is that then we can kind of enumerate very easily in this way how many reactions are there. Recall that there's one, right? That the entire pharmaceutical industry is using one to unite an ethylamine and a propanoic acid, but through combinatorial explosion, there's actually 56 million possible ways to bring these 2 simple pieces together. 8 by 8 grid gives 56 million possible ways, and the entire pharmaceutical industry is thinking about one right now. And each one of these will give a different set of properties, a different shape. Imagine the opportunity in personalized medicine to kind of hit like a chemical structure that fits just the right way. They don't exist yet because they're hard to invent. Like, I mean, it takes me about a PhD student to make one reaction right now. We're trying to automate, we're trying to learn faster. But it's really, really hard to invent a new chemical reaction. And so that's why we've kind of gravitated to the one that works. It works really, really well. Here's an example of one of these new reactions. It's kind of a crazy shape of carbon cages. And it's hard to imagine that we'd ever find a catalyst that could actually turn this and this into this chemical structure. But maybe my granddaughter will be able to talk about it one day. And so we've gone beyond amines and acids. And so this is bromhexine is one of these COVID drugs that we also looked at using this technology. And so with bromhexine, we were able to disconnect from the target molecule, break it down into some simpler pieces that are not amine and acid. This is an amine, and we were focused on the aryl halide bond here. And actually, in the lab, we were able to invent this new reaction quite quickly. So that we take this aryl halide and something that's used in the rubber industry and is dirt cheap and connect it together into a drug that was under investigation for COVID. So the cool thing here is that the starting materials that we use are cheaper than what's in commercial production for this drug. It's just that we had to invent a new reaction, and our reaction conditions these are not very industrially friendly yet. You have to optimize a lot on that. But the cool thing is that by inventing a new reaction, we were able to make a really high impact step. The existing procedure for making bromhexine requires 2 or 3 steps and much more expensive starting materials. So this is kind of the value of inventing new reactions in this way. Dan, how am I doing on time? 20 minutes. Oh. Oh, wow. All right. We can like take this in like a meal. All right. Okay. So one of my big drivers in leaving the pharmaceutical industry and coming to be an assistant professor was to get back to my roots in total synthesis. This is like my favorite thing to do, is to make really complex molecules just because we can, and think about how we could break down molecules into pieces and actually go into the lab and do it. And when I trained in organic chemistry, it was quite routine in those days, in the early 2000s, that teams of 20 people would work 25 years to make one molecule and just say, we did it. There's value in learning how to make complex molecules. Right now, as I showed you, we use the amide coupling as this really trusted, robust reaction, as a single reaction to make drugs, and that kind of influences the way that we think about drugs. We might make something that we know could be better. We might end up launching a drug that's a 200 milligram pill, when if we worked on it a little bit harder, if we were more brave in the synthesis that we embarked on, we could get it down to a 20 milligram pill. You always want to give your patient the lowest dose that is possible. But we kind of, we have to compromise, right? Because you have to design what you can make quickly. The pharmaceutical industry doesn't have 20 years to invent new reactions and think about reactions that don't exist. And so, anyways, total synthesis is an art. It's often kind of alluded more to art than science, or it has a lot of artisanal qualities to it. And how do you stitch together molecules in such a way? And so this was a molecule that caught our attention. I'll tell you how we tackled it using this SYNTHIA software. And again, in full disclosure, I am sponsored by Sigma, who sells that software. And so this is STEMOAMIDE. It is isolated from the stemona plant, or baibu, which is a Chinese traditional medicine that's been used for about 300 years for afflictions of the lung. And so stemona plant is associated with the lung qi, whose purpose is to harmonize how to live as a spiritual body in an animal body, which to me is a great opportunity to embark on a total synthesis project. And one of the cool things was that it had been benchmarked with 32 total syntheses. So there's 32 papers out there on how to make this particular molecule. They range in step count from 26 steps down to 7 steps. And so we had this, the cool thing was that you wouldn't do this if you were writing to the NIH or the NSF and saying, I want to make a molecule because 32 people have done it before. But because we had an industrial sponsor, they wanted to have something that we could benchmark their software against. And so actually the fact that this had been made many, many times before was attractive for us. And as we worked on the synthesis, there were 26 syntheses when we started. As I said, we've been working on this molecule for 4 years, and so now there's 32. And they range in step count, number of reaction steps, from 26 down to 6. And so we plugged STEMOAMIDE, shown here, into the SYNTHIA software. And again, this is the software that has these 100,000 rules that were encoded by 15 years of Polish PhDs. And it understands which reactions are going to play nice with each other, and which things are going to need to be protected because the catalyst that you need for that reaction is going to poison the functional group that's in there. And so it understands these types of rules, and it understands which chemicals you can buy. But then within that kind of box, there's this combinatorial explosion. And so you need a computer to kind of navigate through that combinatorial explosion and find the pathways through. And so shown here is STEMOAMIDE is the yellow dot in the center, and a sequence of 50 different routes that the software gave as an output, shown as this network here. And so they terminate in these pink dots, which are the commercially available materials. There's these purple dots, which are the intermediates along the way that we would need to characterize, isolate, and purify them, and take an NMR or IR, a mass spectrum of. And that's the experimental work that we go into. The cool thing about this particular answer that the computer gave us is that it was invoking this organocatalyzed Mannich reaction in every single one of its answers. Do you see this orange tetrad of dots? So this is called the Mannich reaction, and it was actually a proline catalyzed Mannich reaction that it invoked. So recall that over the span of, almost 3 decades, 32 groups have made this molecule. Not a single one of those 32 publications uses the organocatalyzed Mannich reaction. So you might think, well, maybe the organocatalyzed Mannich reaction is like rare, or it's like, it's kind of hidden in the literature. And so that's why no one used it before. But it won the Nobel Prize 2 years ago. So it's not like it's hidden. We hadn't thought of it. None of the other 32 groups that had published in this area had used it. And so we thought it was kind of cool that the computer invoked it every single time, right? And so this was kind of neat. Now the shortest route in here, I think, is this one, and it was 7 steps, which was competitive with the shortest published route to the molecule, but it wasn't better than. And so we thought that, all right, we'll take it, we'll work with it because the Mannich reaction is kind of that it's invoking this, but we're not really shorter than the state of the art. We're competitive with, and yeah, we got a little lucky. So Dr. Yingfu Lin went into the lab and he developed this protocol. So here is the organocatalyzed Mannich reaction as our first step. We take 2 equivalents of this aldehyde and this amine, and they stitch together using L-proline as the catalyst, and it makes the organocatalyzed Mannich. Now we got lucky in that the next 2 steps the computer proposed actually happened in the same pot. And so they're shown here, where he quenches with allyl zinc bromide. That snaps into this aldehyde, and then the alcohol that comes from there closes onto this and forms this ring. And so in a single reaction pot, we're building up the bulk of the complexity of this molecule very rapidly. There's some zinc salts that are floating around at the end of this reaction, and so he filters off all of those zinc salts and then treats with a very strong acid called TFA. But this is very operationally simple in the context of a lot of organic chemistry protocols, because we don't do any purification until we get to here. So we buy these, and then we purify here. And so that's like streamlined a lot of the route. And so we have a lot of the stereo centers. These are the things that are hard to build. Hopefully you can see that there's like a dashed H here and a wedged H here. And so those are hard because they're 3-dimensional. And we're getting a 38-to-1 ratio of the different combinations of these stereo centers, and a 99% enantiomeric excess, which is really good. And so we were really pleased with this experimentally. And so in 2 steps, we had built a lot of the complexity of this molecule. And then we need to do what's called the end game of the synthesis, where we want to go from here into the final product, which is this guy here. And to do that, we go through what's called a hydroboration oxidation, and we install this alcohol. Then we do what's called an Appel reaction, which takes the alcohol into this bromide. And then we do an SN2 reaction. If you've taken ORG1 or ORG2, you maybe have seen the SN2 reaction. So it's just, this is the nucleophile, and this is the electrophile, and it kicks out this bromide, and you make the 7-membered ring. And then we install this methyl group, and this was actually known from the 32 existing syntheses that you could stick this methyl group on as the last step. So we knew that. And so using the computer and finding this Nobel Prize winning reaction from a combinatorial explosion of possibilities, we achieved a 6-step synthesis of the route. We got a little lucky because 2 of the steps ended up experimentally stitching themselves together. And so we had predicted a 7-step route. In practice, we got a 6-step route. And so now we had the shortest synthesis of this molecule. And so that was exciting. But as organic chemists, we were not satisfied. We're rarely satisfied. The product of this Mannich reaction was really satisfying, that we got there quickly. But these 4 steps we were a little frustrated by. Here I'm doing a hydroboration oxidation to install this alcohol. And then I turn this alcohol into this bromide. There's no alcohol in my final product. There's no bromide in my final product. These are things that I had to do experimentally to get to the end. But it's kind of like wasted effort, because I've installed an alcohol, and I don't, at the end of the day, want an alcohol. I've installed a bromide, and at the end of the day, I don't want a bromide. And so maybe we could try and find a way to understand this and streamline this a little bit more. And so I want to go back to these graphs. I apologize that the color scheme of this graph doesn't map with the exact color scheme I showed earlier, but that shouldn't matter too much. What we found is that these graphs can be used, or these adjacency matrices can be used to map the entirety of a total synthesis route. And this was a really fun discovery. So here on the right, we have, or my right, your left, we have the final product, and it's got 16 heavy atoms. And then there's a bromide, which is atom 17. There was an alcohol, which is 18, and there was an ester that we kick out the methoxy from, which was 19. And then there's a protecting group, which actually has quite a few atoms, but we only care about the carbon that's stuck onto our thing, so we'll call that 20. So we have 16 heavy atoms of the product and 4 other things that go into the mix. So 16 plus 4 gives me a 20 by 20 grid. And what I can do is I can track all of the bonds for my entire total synthesis in a 20 by 20 grid. I can make, I have a 6-step synthesis, so I have 7 adjacency matrices, and they're all 20 by 20. And what I can do is I can take the final product, and I can look that adjacency matrix is to the product before it. And so hopefully you can see that this graph or this matrix kind of looks similar to the final product. And similarly, the penultimate intermediate looks very similar to the final product. Now, if I go all the way back to the starting materials look less like the final product, and their graph looks less like the final product graph, right? So we can measure the similarity as we go down. And what we see from this route, so if I make the 7 graphs of this route and I subtract or look for graph similarity among them, recall that we have a really nice key step at the beginning, which is this Mannich reaction, and then we have these kind of less impactful steps where I install an alcohol, I install a bromide, I do an SN2. And so the graph similarity looks like this, where you see the key step right away, right? It's like I have like a big impact and it was successful, and then I kind of like drag my feet for a little bit. And so we think that this is going to be really an impactful way to streamline chemical syntheses, to necessarily invent new reactions from this. And so what we would love to do, you see how there's like a little positive inflection here? If you actually draw a line from this dot to this dot, we would get a 4-step synthesis, and it would involve a reaction that doesn't yet exist. And so we're trying really hard to invent that reaction. We're hammering it with our robots. We've had a really difficult time making this one particular reaction. We can't find the catalyst that's going to make it work, but we will find it. We're going to keep trying to like streamline this route. But the cool thing is that we now have like a system to figure out like which steps are good and which steps are not good, and let's streamline around those not good steps. So when we did this synthesis, we were kind of following what was happening in the literature. We're not the only team that's working on automated retrosynthesis or even using this SYNTHIA software. Many people are doing this. And typically I think that the conversation that's happening in the literature right now is like, is the computer as good as a human? And if the organic chemistry community does not believe that computers are going to, they don't want to talk about how they're going to have to compete with computers. And so it's all about like, here's what the computer said and here's what an organic chemist said. There's like a Turing test paper that was in Nature last year where they did a whole bunch of synthetic routes and in the computer and by humans, and humans can't tell if something is made by the computer, for instance. But it's all about like following the computer's recipe, right? That like, here's the answer the computer gave and then I'll follow it. And that's what we tried to do here. So we tried to stay as true as we could to the computer's recipe. But now thinking about this, like the cool thing that we liked about this was we found a key step, and then we kind of followed the computer's recipe to get through the rest. What if we have this ability to navigate through combinatorial explosion because of the computer and these 100,000 reaction rules that were encoded by 15 years of Polish PhDs? What if we like generated thousands of routes and we looked through them, finding key steps from this route over here, and key steps from this route over here, and key steps from this route over here, and tried to stitch them together. And what we arrived at was this sequence. So we buy this lactol, we do a brown allylation to install this allo group. We then do what's called an Ender's Michael addition with this cyclobutanone, and it stitches in, we quench with methyl iodide, and we've got this kind of interesting intermediate, which we're going to do a hydroazidation. And so we do an anti-Markovnikov hydroazidation. We work up with TICL4, and the sequence is complete in 3 steps. And so this was a very exciting day in the lab, and we were able to take the shortest synthesis of this molecule and then slice it in half using this approach. And so our graph edit distance for this 3-step sequence looks like this. You can see that there's a high impact to each of the steps that we're engaged in. We've pushed this molecule forward using more classic organic chemistry techniques, following some of the work that was published earlier by Chita and Sato. And this molecule is being tested in the hospital right now with some mice who have sick lungs. And so we're, hopefully, normally I talk to organic chemists. As you can see, I'm very excited to talk more with data scientists. And you can see that there's, 200 years of organic chemicals being thought of as graphs. And there's a huge opportunity for merging with data science. As I said, we're about to put out more chemical data than is out there right now, which is about 200,000 data points, and it's going to be about 1 terabyte of data. This is like going to be way more than has been released in organic chemistry before. And so my team is really incredible to work with. I think these funders, I think my wife, our school is closed today, so she was able to support me coming here with our daughter at home. Thank you so much for this opportunity.
We have time for maybe 1 or 2 questions. Do you have some questions? There's one back there.
Hi, thank you for the interesting talk. I'm Cameron. I'm a doctoral candidate in chemical engineering, and I do research on aqueous flow batteries using artificial intelligence. Organic aqueous flow batteries, excuse me. So this, I think the idea of automated synthesis, like catalyst synthesis and testing for organic reactions, is very interesting. From an organic chemist perspective, do you feel like, and I don't know how close you are to this field of work, but do you feel like uncertainty quantification is a necessary and important challenge to automated synthesis of these chemicals? Because the space seems so combinatorically large, right?
Yeah.
So.
Yeah, thank you for that question. And so absolutely, I do think that uncertainty is a major part of where we are as a field. There's just enormous technical challenges which are totally overcomable, but they just haven't been overcome yet. I mean, the challenge that we have is all of our chemicals need to be handled away from air and water. And so we've figured that out. We put glove boxes around stuff, and you can kind of work in that space. But then another thing is, like, things only want to be handled at very low temperature, and it's kind of hard to cool and then later heat in that environment. So there's a lot of engineering that is all coming online. It's all totally solvable problems, but I think that we're kind of in this space right now where there's a lot of vendors who are selling these pieces. There's no standard quite yet. Hopefully in a couple years that will be there. And we run 96 reactions and we get a lot of kudos for it. We're able to run 1,500 reactions, and that's we're one of very few labs in the world that can pull that off. But it's not that much, right? I mean, 1,500 is really not that many data points. And so we are playing through uncertainty in that space, but I would say that it's a little sad and embarrassing where we are as a field right now.
Okay, we're going to take a break in a second so you can have some questions offline. I just wanted to make an observation that one thing that occurs to me is that the trends that we're talking about this afternoon provide greater and greater capacity for agile response to emergencies and unknown things. I mean, you saw flavors of that in the COVID response, and you'll see flavors of it perhaps in what UK is going to talk about later. So I would encourage everyone to think about, as they're designing environments for doing the kind of standard academic research, think about how they could be deployed against unexpected external forces and so forth. And I guess, I mean, this among other, the work you're doing among others was probably instrumental to this incredible response and short delay we got relative to the past in getting effective vaccines. Also, thank you very much, and we're all looking forward to the Nobel-winning Cernak process one of these days. Thank you very much. All right, we're going to take a 10-minute break. It's